Thanks very much, Mark. Thanks for everyone for coming. It's a real honor to be speaking at the evenings at Whitney. I'm always excited to be out in the audience and watching what's going on, and excited to see everyone that comes out. So I'm going to talk about some of the work that we do on DNA sequencing and how I see animal history and animal evolution through the eyes of someone who just looks at DNA mostly. Not just looks at DNA, but looks at a lot of DNA. Okay, and so I'm going to start, since I'm fairly new in the community and I know a lot of you by sight but don't know you very well, I'll start by talking about how I got to the Whitney lab, and then I will switch to a quick, gentle primer on genomics and evolutionary genomics to bring everyone up to speed in case you know a little bit, but maybe you could use a little bit of reminder of what it all is about. And then I'll talk about two projects that we've done at the lab here at Whitney. One is sequencing an earthworm genome, and the other is on ctenophore reproduction. So my path to science is actually, you may see a scientist give a talk, and they'll say, I was 4 years old, and this ant crawled across my nose, and I was like, I know I want to be a biologist, right? But I was 24 years old, and I had no idea what I was doing, right? So the only thing I knew really is that I needed to get a degree. That's all I knew. That's basically all my parents said is get a degree, whatever you do, you'll figure it out. So I went to all these places to try to figure out what I wanted to do, and I stopped here. I finally went back to community college and got an Associate of Arts because I was like, I better get something because I've been doing this spinning wheels for 5 years. And I got an Associate of Arts, and this actually helped me get a job later, so I was glad I did it. And then I was at a bar, and I was talking to a friend of mine, and we were just like, yeah, no. She's like, I just started this program. I'm studying marketing at University of Baltimore. It's great. I was like, all right, I'll do that. So I went to the University of Baltimore, and I signed up for information systems. And there, it was cool because I started doing computers, and I was like, I like computers. And I met this guy who, in my class, the teacher said, if anybody has a question, go to this guy. His name's Ali Seji. If anybody has a question, go to him, and he'll answer your question. So I was like, I got to be friends with that guy because he knows a lot of stuff. So I became friends with him, and then we started this company in '95 called Quantum Web Design. And to be a web designer in 1995 was a good time to be a web designer. Right now, probably you have a lot of competition, but then we could do whatever we wanted. And the only thing, we were pretty good at coding, but we were terrible at getting money out of people. And people don't really, surprisingly, will not pay you unless you ask them 3 or 4 times, and we would only ask once. So we never got paid. And I was like, okay, now I know I like to code, and I know that I don't like to be in business. So I said, let's get a job. I went and got a job, and at the time there was no Craigslist, but there was this thing called Usenet, which was basically like Craigslist. So I basically went on Craigslist, and I was like looking for jobs. I saw this job for programmer, and I went and sent my resume and was like, okay, I want to be a programmer. I've done some things, and I ended up at the National Institutes of Health. How great is that? I had no idea what that was about at all, but I could go there and be a programmer, and it was great. This was a new institute, and my boss was this guy, Andy Boxavanis, and I started making calendars and address books and little web pages, and then the data started pouring in. This was a new institute, and they had lots of data to process and not that many people who knew how to program, so they were like, hey, do you want to look at genetic data and genomic data? I was like, sure, I'll do that. And next thing you know, I'm working with these world-class scientists. Everybody's got PhDs. I don't even know what PhD stands for. And I'm hanging out with these people, and I'm like, okay, these are normal people. I could do this type of thing. And I kind of fell in love with doing research. So while I was at NIH, I was taking night classes. I had switched to another school, imagine that. And I got my bachelor's degrees in computer science while I was working at NIH. And as soon as I got my bachelor's degree, I'm like, okay, now I'm going to go get a PhD so I can do my own research. And I went to Boston University, as Mark said, and they had just started this bioinformatics program. And this was perfect for me because I didn't know very much at all about biology, but I knew a lot about computer science, and I didn't know very much about any of these things. So I could go to this school and basically fill in all the gaps in my knowledge that I needed to do, and that's what I did. And the other thing that was nice about this program is I didn't have to know what I really wanted to do when I got there, because again, I didn't know. And they had this idea that you would rotate through labs. And I wanted to be in biology, so I looked at the faculty of biology, found this guy, Chris Schneider, who had a snake, and he was holding it. I was like, this is tough. Maybe I should go work with this guy. So I went into his office and talked to him, and he's like, yeah, you know, you have some skills that are interesting, but not for the stuff that I do, but there's a guy 2 doors down the hall who I think you would hit it off with really well. And there was this guy, John Finnerty, and he studied sea anemones. A little weird for me. I was like, snakes? Snakes seemed cooler than sea anemones at the time. Now I totally think differently. But we hit it off, and I talked to people at NIH, and I was like, hey, this guy, clearly we get along, but he studies sea anemones. What's up with that? And they were like, if you get along with your advisor, that's the person you should choose. And I took that advice, and I would give that advice to somebody. So your mentor is important, that you really click with them. And we clicked, we did some great work. And then I went back to NIH because I had enjoyed working for Andy, and I went back and worked in his lab, and I said, hey, I've been working on these weird animals. If I come back, could we do weird stuff like this? And he was like, yeah, let's do it. And I think he's glad he did. We actually were collaborating with Mark a lot, the director here, and when he was in Hawaii, and he was an expert on these animals, and we were like, let's sequence this genome, and we came up with some really good, really exciting studies on this guy. And then I had a little bit, I was like, you know, I don't really want to get a real job yet, and I'd like to go to Europe. It'd be great. So I did another postdoc in Bergen, Norway with Andreas Heinel, and you could see the weather in Bergen was fantastic. We had rain, we had wind going this way, and it was cloudy all the time, but it was a really fantastic experience. And that's one of the nice things for all you younger folk out there that are thinking about what you want to do. Being a scientist is cool because you can live in cool places, you can make friends all over the world, and you can work on really weird animals. So this guy, Andy, he works on the crazy, like all animals. We were in his lab. We had all these animals in the lab, and I didn't know anything about these animals, and that really helped fill in my knowledge about zoology and the breadth of diversity in the animal kingdom. Okay, then I ended up this place, and I really hit the big time because, I mean, the Whitney lab is a fantastic place. We have great faculty, we have great facilities, we have a community that you can see is very supportive, and it's a beautiful place to work, right? So I started my lab, and my lab is centered around evolutionary genomics. So in order to understand what I mean by evolutionary genomics, I'm going to start by just taking you through what is a genome, the basics. When I think about genomes, I think about animal genomes, but there's plant genomes, there's bacterial genomes. But when we talk about multicellular organisms, like animals, they're made up of cells. Everything in our body is made up of cells, right? And each one of these cells has a nucleus that has chromosomes here, and these chromosomes are the DNA. This is our genome right here. So every one of our cells has a copy of the genome, and it's the same copy in every cell. So we can sequence one of these. Oh, let's get into a little bit more. So what is a chromosome? It's millions of these chemicals: adenine, cytosine, thymine, and guanine, A, C, T, and G. And there's millions of them that are in this stretch of chromosome. So we can, so one of the things we do is, there's a lot of biology, there's cells, there's all this thing, but we can really just abstract everything into ACs, Ts, and Gs, all life into ACs, Ts, and Gs. Sounds horrible, but we can find cool things and then take it back into biology, and that's what we do. And when you look at these ACs, Ts, and Gs, there's also patterns in these things, and patterns in these letters. And for example, this would be a gene, and there's a certain code, the 3-letter code that marks the beginning of a gene, and a certain code that marks the end of a gene. And there's even more complicated things inside of these genes. But And genes make up about 1% of our genome. And other animals, it's a little bit different, but when I study, the way I like to study genomics is I just kind of ignore all this other stuff. Other DNA between the genes, I concentrate on the genes. Other people love the stuff in between, and I say more power to you. But the animals I'm studying, it's much easier to pull out the genes, and there's so much information about the genes. Okay? So then what about the evolution? So since I started doing research, really at the base of my question is evolution, and is the ideas that Charles Darwin put together in the 19th century. So if you combine genomics and evolution, what kind of questions can we ask? And the kind of questions we can ask is, if we look at this gene, it's a blue gene, right, in humans, and then we look in these other related primates for this gene, what might we see? Well, we might see that each one of these primates has that same gene, and it all has one copy, and they all have the gene, right? And then what could we infer? We would infer from that the common ancestor had that gene, and that each one of these lineages inherited that gene. But we might infer things like gene loss, where we have this green gene, and that every one of these other primates has the green gene except for gorillas. And then what we would infer is that the common ancestor had that gene, and all these other lineages inherit it, but this gorilla has actually lost the gene. And why would we care about that? Well, if we know the function of that gene, and the function of that gene has a biological relevance that maybe, for example, let's say it's a gene that's involved in breaking down certain types of sugar in metabolism. Let's say gorillas can't eat that type of sugar. Well, it might be explained by the loss of that gene. It could be that simple. It's usually more complex than that. But knowing animals' genomes and knowing where evolutionary events like gene losses occurred are important. You might look at this and say, well, this is one explanation, but I can think of that in a second. Hold on. Let's talk about gene duplication. You might have one gene in humans, and you might look across on all these primates and say, here in the gorillas, you have 2 copies of that gene. Well, what that would infer is that the common ancestor had one gene, and then that there was a duplication event in the gorillas. And again, if we know the function of that gene, and we know the biology of the animals we're looking at, there may be some way to make an inference about what's going on there. But then you might say, I said it too early last time, you might say, well, that's not the only way you could get this pattern. You might say the ancestor had 2 of these genes, and then it was lost right here. And that would be a good thing for you to call me out on. But I would say the way we do these types of analyses is we try to come up with the most simple explanation for the data. And that's how we have to work, because we can't go back in time and sequence ancestral genomes. We have to use Occam's razor or whatever, parsimony. The most parsimonious explanation is that this explanation of one event versus this explanation of 5 events, we have to go with one event. That's the best we can do. It's the most probable thing that's going on. Okay, so these historical events drive biodiversity. We know that genes duplicate and genes are lost throughout evolution, and these are all very important things that happen. And we know, and if we look at animal life, there's so many things that are happening, right? And a lot of these events are really driving this diversity that we see. Oh, I'm going to be in trouble here. Okay, so now I'm going to switch gears to the earthworm genome, if it behaves. Okay, so the earthworm genome is a project that we started, and really it was driven by this program that was started here at the Whitney lab in 1987, which we call REU, Research Experience for Undergraduates. And this is an NSF, National Science Foundation, funded program where we have undergraduates, usually from institutions where they don't have research opportunities. They're at smaller liberal arts type schools or community colleges. They come to Whitney, they live in the dorms, and they spend 10, 11 weeks here. We usually have 8 students, and they work with a faculty member in that faculty member's lab. And this program is really awesome because we have the longest running REU program in the country, thanks to Barbara Battelle, who started it in 1987, kept it going for more than 25 years. Barbara is an emeritus faculty here. And then she, in the last few years, passed the torch to Elaine, and she's kept it going, and we recently got it refunded. And so hundreds of students have come through the Whitney lab and spent the summer and went back out and went and done and made careers in science. So I think it really makes a difference. And if you're a high school student or an undergraduate in the audience, this is something that you should keep in mind as an opportunity, because there's over 1,000 of these programs across the country. You can go to Hawaii, you can go to Harvard, you can go to Alaska, you can go to Texas, you can do whatever you want. You can study engineering, you can study math, you can study computer science, all these availability. So it's a pretty amazing program. And for me, it's amazing because I usually get a really great student who works hard, and I'm going to talk about a program by Allison Zwarek, who was in my lab for the first summer, and she was interested in computational biology, and I said to her, okay, well, you have a project. This paper in 2013 had just come out. This is, Allison joined my lab in 2014, and this paper came out. And one of the interesting findings of the paper, they had sequenced the genome of this polychaete and the genome of this leech. And one of the cool things they found is that the polychaete genome was evolving very slowly, and that the leech genome, on the other hand, was evolving very quickly. So you might ask, how do you know if something's evolving slow or something's evolving fast? Well, one of the metrics that's used is the number of gene duplications and the number of gene losses. So I just showed in the primates how you would infer gene duplications and gene losses, and they did those types of analysis in these marine worms. Or these are not actually marine, but anyway. What they found is very few gene duplications and gene losses in the polychaete, and many, many gene duplications and gene losses in the leech. So the question I was interested in is, how do evolutionary rates change? Why is the polychaete evolving so slowly, and why has the leech gone completely crazy? But the first question you have to ask before you can address this question is, where on the tree do the rate changes occur? Okay? And this is the question that I had Allison ask. And the way we address that is we sequence the genome of this earthworm, and we could say, there's two places that this leech speed up could have happened on this tree. One is that it happened here before earthworm and leech split, right? And if that was the case, then what you should expect is all the losses and duplications that you see in leech should be present in the earthworm. Excuse me. So that would, because there's shared evolution here, you should see losses and duplications in both. But if it happened that the losses and duplications occurred in the leech and that the evolutionary speed up occurred here, then what you would expect in the earthworm genome to look more like the polychaete, right? That there's not a lot of duplications and not a lot of losses. So it would be specific to the leech. So we sequenced the genome of the earthworm with our collaborators at Rice University. And the way you sequence a genome, it'd be nice if the technology would just start here and go, but it doesn't do that. The technology currently, especially in 2014, was kind of, it's getting better, but really you can only sequence chunks at about 400 base pairs, 500 base pairs, 508 letters A, C, T, and G. So you kind of have to zap the whole genome and create these small chunks and then sequence these little bits. So that's what we did. And then Allison used a variety of algorithms to put them back together as well as she could. And basically, it's like a puzzle. There's overlap, and you go out as far as you can go, and you try different algorithms and use different metrics to decide which one is the best. So Allison did that in about 3 or 4 weeks when she first got here. So just to compare assembly quality, the leech genome was sequenced by a huge team at the Department of Energy's Joint Genome Institute, and so was the polychaete genome. And they worked on it for about 5 years. And one measure of quality is the number of pieces that the genome is assembled into, because you have about 20 chromosomes in most animal genomes, give or take a few. So this genome was assembled into 2,000 pieces. The ideal would be 20 pieces, one sequence for each chromosome. But 2,000 means that each chromosome is in about 1,000 pieces. But that may sound bad, but given the technology, this is amazing. This is fantastic. This is a great genome. Even 20,000 is pretty fantastic, right? Especially when you see the number that we have. We have 1.7 million, which is terrible, right? It's okay. The cool thing is 1.7 million sounds bad to probably most of you, and it sounds bad to most genome biologists, but I was convinced that we can take a very rough genome and find some very cool things, and I think I'm going to try to convince you that we did that. And the reason we can find cool things is because we're interested in the cool genes. And these genes, homeoboxes, are pretty short. The domain, the important domain, which binds DNA, is coded for by 180 nucleotides, okay? 180 A's, T's, and G's. And if our sequencing chunks are 400 or 500, then we should really be able to find all of these. The other cool thing about homeoboxes is they're crucial for development. So these are like bosses. You know, they say, okay, I'm going to go turn on this gene, this gene, this gene, this gene, and we're going to go make a leg. And I'm going to turn on this gene, this gene, this gene, we're going to make a wing. We're going to make a brain right now with this gene, this gene, this gene, this gene. So if you mess with these genes, if they get mutated or they change, most likely the animal doesn't survive. So they're highly, highly conserved. That means they're easy to find in genomes. And they're part of a large family. So humans have about 333, and you see 100 in fly, 100 and few in the oyster. So we looked in the earthworm, and we found 440, which is pretty remarkable. It's even more than human, which usually has more genes than most things. And the one thing you might be thinking is, especially if you're a genomicist, is that if you have a crappy genome, then maybe this is just an artifact that there's so many homeoboxes because the genome's not well. And I can't really go into how we proved that that's not the case, but our reviewers, this paper was eventually published in 2015, our reviewers had the same concern. So we went about showing that and convinced 3 reviewers who were knowledgeable in the field that this is really, we really have 440 genes. But one thing we wanted to do is also run our method, which tends to be a little bit more sensitive, I think, than some of the older studies. And we found additional homeoboxes in at least the polychaete and the leech. So this was important that we're comparing apples and oranges when we get to the next stage of the analysis. So right out the gate, Allison, in probably the first 6 weeks, had assembled a genome and characterized 440 genes from that genome. Pretty exciting stuff already. But the really exciting stuff is when she started to look for duplications and losses in those genomes, in those homeobox genes. So what I'm showing here is going to be a bar graph, and each one of these letters is a class of homeobox genes. So I took the names off because they're crazy, and then you would think, I got to remember these names, but you don't need to remember them. And I'm going to show all of the different lineages and their duplications. So the polychaete lineage right here, we see very few duplications that occurred. This isn't surprising because this is what was published in 2013 in that previous study. This is a slowly evolving genome. Losses and duplications are not occurring that much. This genome really looks very similar to the ancestral genome. But if we look at this lineage here, the stem of the leech and the earthworm, we actually see a lot of duplications. Not that many losses, but many duplications. So we would say that the speed up of evolution really was already occurring at this stage. What if we look at the leech bit? Well, the leech lineage is pretty interesting because you have this crazy bar right here, 31. That turns out that that's one gene that's gone crazy. That's duplicated, which is an interesting evolutionary event. What is that one gene doing in that animal? It's CDX, if you know the gene. It's an interesting developmental gene, and it's gone crazy in the leech. But all the rest of the families have very few duplications, but then you see quite a few losses, more losses than you see anywhere else on the tree. So this lineage is kind of a weird one. And then you look at the earthworm, and things have gone crazy. So the speed of evolution was pretty fast here, but I think that it's really taken off after the earthworms split from leech. And okay, so just to wrap this story up, the significance of why do we care about this stuff? I mean, one is it's history, right? We care about history. We care about what's happened in the past. And so we've kind of documented some interesting genomic events that have occurred in the past. Also, these animals are interesting from how they're related. They interact with us. From earthworms, if you're making a garden, earthworms are your friends, right? The more earthworms you have in your garden, the better your soil. And they're good for composting. They are good for, they can deal with metals. So if you have an area of land that's been exposed to some copper or metal and stuff like that, you can throw earthworms in there and they'll clean it up for you. So how that works is interesting. And since we've provided a genome, we have this earthworm genome resource, people can start to look at that. And we have a better idea of how their genome is evolving. And then leeches are interesting from a biomedical standpoint. They're still used for microsurgery. So anything we can glean about the history of the leech and the evolution of the leech lineage is going to be interesting as well. And so we've placed these evolutionary events on the tree, and this will be important to really explain the mechanisms that are driving evolution in these lineages. And lastly, we've taken a very drafty genome and really made some interesting biology about it. So that kind of opens up the possibility of sequencing things fast and, you know, getting an army of undergrads and finding like everything about everything. Okay, and it gives us some insights on how biodiversity occurs. So the very cool thing about this project too is that Allison went on and she started a master's degree in bioinformatics in Troy, New York at Rensselaer University, and then has recently just been accepted into a PhD program in Wales, in the UK. So this has had a major effect on Allison as well. Okay, I'm going to switch gears. And everything I told you about evolution and genomics is really doesn't matter anymore. So you can throw that out. I'm going to, and the reason I'm kind of switching gears is because I want to tell you a cool story, and we have a cool project that we're working. And I also want to show you that our lab is not really just about sitting in the computer and looking at ACs and TGs all day. We do other things. So this is work that's being led by a postdoc in my lab, Daniel Sasson, and it was also involved an REU student, Anya Hakes. And we were interested in reproduction in ctenophores. And just to step back, ctenophores are these animals. Most of the animals that are showing here are about the size of a baseball. If you look right out here in the intracoastal, there are ctenophores out there now. I know they're out there because after the hurricane, I actually had them in my pool, which was pretty cool. When your study animal shows up in your pool, it's pretty amazing. And the thing that's interesting about their reproduction, one is that there hasn't been a lot of studies on the reproduction of these animals. They're tough to keep in the lab if you don't work at a marine lab like we do. They're not great lab rats. Lucky for them. And the other is that they are simultaneous hermaphrodites, so they're both male and female at the same time. And we were interested in how do you stay a simultaneous hermaphrodite? So almost all ctenophores are simultaneous hermaphrodites, and they're able to self-fertilize. So how do you maintain that? One of the big questions Daniel wanted to know is there effect? Is there an effect of self-fertilization on viability of embryos? Because we know that when humans have children with their close relative, these children suffer from inbreeding, and the level of developmental problems, including mortality, are pretty great when this happens. And if you're self-fertilizing, that's like inbreeding on steroids. So the first thing was the first experiment he wanted to do was he took animals, he collects animals, and if you bring them in and put them on a benchtop, overnight they will produce, they'll spawn and produce eggs and sperm. So he took animals and he put one animal in a dish, and he would do that for like 30 animals, and then take 2 animals and put them in a dish, right? So you have alone and you have paired samples. And then just count, first count the number of eggs that are produced, and then what percentage of those eggs in 24 hours. So you count, the next morning you come in, count the eggs, and then 24 hours later he would count the number of eggs that have developed. And you can see from this that the animals that were alone produced much less of the eggs were developed than the animals that were spawned in pairs. And this suggests that there is a cost to self-fertilization. But the cool thing in science is when you're looking for one thing, something else might pop up. And that happened because since Daniel was counting the eggs, he noticed this pattern where he's getting basically the same number of eggs, whether you have 1 animal or 2 animals. And you would expect if you have 2 animals, you'd get twice the eggs, right? So since he's a behavioral ecologist and a reproductive specialist, he knew about these there's some animals that are simultaneous hermaphrodites that will actually, snails and some fish, that will talk to each other and say, hey, I will do the eggs, you do the sperm, right? And so he hypothesized that maybe we're seeing these numbers because the ctenophores have some similar system. In order to do that, we couldn't really just count the eggs in these guys because all the eggs from both animals would be going to the same dish, so it would be hard to tell who's who. So we built tennis courts for ctenophores. And actually, we got the guys in the shop, Adam Passetti and his crew, to build these tennis courts. And they're basically plastic aquaria that have a plastic divider. And we seal that divider along here. We sealed these pretty large holes and drilled, if you can see, these really tiny holes. And the idea was to have holes where chemical cues could go through, but that eggs would not go through. And we tested if you put one animal, do the eggs get through? And we get very few eggs going through. But the idea was, Daniel's hypothesis, that one animal would produce a bunch of eggs and the other would produce sperm. But quantifying sperm is a difficult task, so we haven't done that. But we're just dealing with eggs at this point. And if the hypothesis was correct, you would see on side B, maybe you get 1,000 and 0 on side A. And you'd always have some on one side and 0 on the other side. But then there's the alternative hypothesis that egg production is just random. So we have some eggs over here, some eggs over there, and they weren't talking to each other, so you just got eggs. And then we got our results, and there was some that looked like, oh, there might be something cool happening here. You know, we have some zeros in column 2, 3, and 8, I think. But then a lot of the columns were like, you know, maybe this is random, right? So how do we figure out if there's anything significant here? Because after looking at it and looking at it and looking at it, we're like, well, you know, these are pretty big differences in some cases, right? Maybe this isn't a random pattern. And the way we tested that is, first we subtracted each side from each side, and we took the absolute difference for each side, and then we added it up into 8,848. And then we built random data sets from this data. We sampled this data randomly, and we build new data sets. So basically, you could take side A, trial 5, and side B, trial 10, and you'd put that as the first one. So we write a computer program that basically will make thousands and thousands of fake data sets that are sampled from our real data and see what were the sum of differences for each one of these fake data sets. And then we can put those in a distribution and see we can see here that about at 5,200 is about the median of this distribution. So really anything that occurs in here, if our number occurs in here, it's probably random, okay? Because if we sample our data, it builds this distribution, so any number that we get would be random here. If the number is very, very small, say less than 2,500, there's probably some regulation happening. And the regulation that's happening is not what we'd expect. It's that there's regulation to produce the same number of eggs. So it says, I'm going to produce 1,000 eggs, you produce 1,000 eggs, right? That's what would be happening if our number was over there. And if the number's over here, there's kind of the opposite. I'm going to produce 1,000 eggs, you better produce like 10, 20, or 100, but no, not a ton, right? And if we look at our result of 8,848, it occurs over here, and we can do the statistics and show that the p-value is quite low. So any p-value lower than 0.01 is considered to be probably statistically significant. And our number is a couple orders of magnitude lower than that. So we feel like this is definitely happening in our animals, right? And if this is happening, it's very cool for several reasons. Because ctenophores communicate to each other, and there really hasn't been any published studies to show that these guys do communicate. Maybe it's not that surprising, but to be the first one to show that is pretty exciting. And that this is a probable mechanism to avoid self-fertilization, so we get some insight into their evolution. How do they keep from this self-fertilization becoming a problem? Also, we can assume, based on this data, there's no mechanic, they're not touching, right? So we have this barrier, they're not able to touch, so we're almost certain that what's happening is a chemical cue. And the ctenophore nervous system is quite interesting, and there's a lot of people that are interested in the nervous system of ctenophores, and it's a bit of a mystery of like what's going on. So now we have a foothold into understanding how ctenophores are able to sense, and we can figure out what that cue is. We have an experiment that we can actually test it and really probe that. So we're very excited to really continue this work and try to figure out how these things talk to each other. Okay, so that's it. Pretty good time, I think. So I'd just like to acknowledge my lab. I have an awesome lab. I've so far hired just amazing people, and I'm super happy about that. I've had really great REU students, great collaborators at Rice University, and all the people at the Whitney lab is a pleasure to work with. And also, you guys, thanks for listening, and thanks for being so supportive.